Hesi A2 Biology Questions. Question 1. During which phase of meiosis does homologous chromosomes separate and move to opposite poles? Anaphase I, anaphase II. Metaphase I, prophase I. Homologous chromosomes separate in anaphase I, reducing ploidy. Anaphase II separates sister chromatids, metaphase I aligns pairs, prophase I does crossing over. Question 2. The sodium-potassium pump moves 3 sodium ions out and 2 potassium ions in using ADP. This is an example of what? Passive transport. Facilitated diffusion. Primary active transport. Osmosis. Primary active transport directly uses ADP to move against gradients. Passive and facilitated diffusion are ADP-free, osmosis is water movement. Question 3. Which cellular organelle is responsible for the synthesis of lipids and metabolism of carbohydrates? Rough ER. Smooth ER. Nucleus. Ribosome. Smooth ER synthesizes lipids and metabolizes carbs. Rough ER makes proteins, nucleus stores DNA, ribosomes synthesize proteins. Question 4. If 2 heterozygous parents, AAXAA, are crossed, what percentage of their offspring will show the recessive phenotype? 0%, 25%, 50%, 75%. Punnett square shows AA, 25%, AA, 50%, AA, 25%. Recessive phenotype, AA, is 25%. Dominant phenotype, AA or AA, is 75%. Question 5. The theory that mitochondria and chloroplasts originated from free-living prokaryotes engulfed by ancestral eukaryotic cells is called what? Cell theory. Endosymbiotic theory. Evolution theory. Germ theory. Endosymbiotic theory explains organelle origins via engulfment. Cell theory covers cell basics, evolution explains species change, germ theory explains disease. Question 6. Which process produces the most NADH and FADH2 molecules per glucose molecule? Glycolysis. Krebs cycle. Pyruvate oxidation. Fermentation. Krebs cycle per glucose, 2 turns, produces 6 NADH and 2 FADH2. Glycolysis produces 2 NADH, pyruvate oxidation 2 NADH, fermentation none. Question 7. A protein's unique 3-dimensional shape which determines its function is known as its what structure? Primary. Secondary. Tertiary. Quaternary. Tertiary structure is the 3D shape from side chain interactions. Primary equals amino acid sequence, secondary equals alpha/beta local folding, quaternary equals multiple subunits. Question 8. Which organelle is responsible for breaking down fatty acids and detoxifying harmful compounds like alcohol? Lysosome. Peroxisome. Ribosome. Nucleus. Peroxisomes break down fatty acids, beta oxidation, and detoxify alcohol/hydrogen peroxide. Lysosomes digest proteins/nucleic acids, ribosomes synthesize proteins. Question 9. The binding of a substrate to an enzyme's active site inducing a conformational change for catalysis is described by which model? Lock and key model. Induced fit model. Fluid mosaic model. Central dogma model. Induced fit model, substrate binding changes enzyme shape for optimal catalysis. Lock and key is rigid, Fluid mosaic is membrane structure. Central dogma is genetic information flow. Question 10. Cells in the human body that are specialized for conducting electrical impulses are called what? Neurons, erythrocytes, myocytes, osteocytes. Neurons conduct electrical impulses. Erythrocytes carry oxygen, myocytes contract muscle, osteocytes maintain bone. Question 11. In the absence of oxygen, yeast cells convert pyruvate into ethanol and CO2 in a process known as what? Aerobic respiration, lactic acid fermentation, alcoholic fermentation, Krebs cycle. Alcoholic fermentation converts pyruvate to ethanol plus CO2, regenerating NAD+. Aerobic respiration requires oxygen, lactic acid occurs in muscles, Krebs cycle is aerobic. Question 12. Which of the following is not a characteristic of living organisms? Ability to reproduce, response to stimuli, constant internal temperature, growth and development. Constant internal temperature, homeothermy, is not universal. Many organisms are poikilotherms. Reproduction, response, and growth are universal characteristics. Question 13. The point on a chromosome where sister chromatids are held together is called the what? Centromere, telomere, chromomere, kinetochore. Centromere holds sister chromatids together and attaches spindle fibers. Telomeres are ends, chromomeres are bead-like regions, kinetochore is protein structure at centromere. Question 14. Which type of cell division reduces the chromosome number by half to produce gametes? Mitosis, binary fission, meiosis, budding. Meiosis reduces ploidy by half to form gametes. Mitosis produces identical diploid cells, binary fission prokaryotic, budding is asymmetrical division. Question 15. When viewing a specimen under a microscope, you see a cell wall, a large central vacuole and chloroplasts. This is most likely what type of cell? Animal cell, bacterial cell, plant cell, fungal cell. Plant cells have cell wall, central vacuole, and chloroplasts. Animal cells lack all three. Bacterial cell wall lacks chloroplasts, fungal cell wall is chitin, no chloroplasts. Question 16. Which enzyme unwinds the DNA double helix during replication? DNA polymerase, helicase, ligase, primase. Helicase unwinds DNA by breaking hydrogen bonds. DNA polymerase synthesizes new strand, ligase joins Okazaki fragments, primase adds RNA primers. Question 17. A disease caused by a misfolded protein that induces abnormal folding in normal proteins is called a what? Viral disease, bacterial infection, prion disease, fungal infection. Prion diseases, e.g., CJD, mad cow, are caused by misfolded prion proteins. Viral, bacterial, fungal have nucleic acids and different mechanisms. Question 18. Which of the following is an example of an anabolic pathway? Glycolysis, lipolysis, gluconeogenesis, synthesis of glucose, cellular respiration. Gluconeogenesis builds glucose from precursors, anabolic. Glycolysis breaks glucose, lipolysis breaks fats, cellular respiration breaks down molecules for ADP. Question 19. The diffusion of water across a membrane is affected by the concentration of solutes. The measure of the total solute concentration is called what? pH, osmolarity, tonicity, diffusion gradient. Osmolarity measures total solute particles per liter. pH measures acidity, tonicity describes effect on cell volume, diffusion gradient is concentration difference. Question 20. In the lac-operon model, lactose acts as an inducer by binding to which protein? RNA polymerase. Repressor protein. Promoter region. Operator region. Lactose binds repressor protein, inactivating it so transcription proceeds. RNA polymerase transcribes, promoter/operator are DNA sequences.